investigator in our field. Ed is, I'm sure, in top 5 most cited investigators in our field. So that it didn't come easy. I'm sure he spent a lot of good hours writing those papers that we all read and all cite. But some people are telling me that all his career resulted from some papers we published together. And I take that as a compliment. So Ed, how many papers did we publish together?
I don't know the answer to that. I should have.
See, this is not paying attention to details. We published 6 papers together. And
it's all about Chris always, Ed. Don't worry about it.
And all of them, some of them I read even, and that's amazing. So with this introduction, let's move on to Sam Du.
Good morning, everyone. Welcome to the Center for Translational Vision Research Distinguished Lecture Series. I'm pleased to introduce Dr. Ed Stone today to everyone. He's the current director of the University of Iowa Institute for Vision Research. He received his MD and his PhD from the Baylor College of Medicine and did his training in ophthalmology and vitreoretinal surgery at the University of Iowa, where he subsequently joined the faculty in 1990 and has remained ever since. He's currently the Seamans-Hauser Chair of Molecular Ophthalmology in the University of Iowa Carver's College of Medicine. He's had a long and very prolific career, publishing nearly 500 papers, contributing to nearly over 20 books, and has been honored by numerous organizations and associations. His career is best known for defining the genetic basis of blinding eye diseases, of which he focuses on macular degeneration and glaucoma, and other conditions such as retinitis pigmentosa and Leber congenital amaurosis. He's been very active in removing the technical, legal, and financial barriers between genetic discoveries and the patients who can benefit from them. He has founded the Carver nonprofit genetic testing laboratory at the University of Iowa that provides low-cost genetic testing to patients in every state of the US and more than 60 other countries worldwide. His current research is in developing affordable gene and stem cell-based treatments for all molecular forms of inherited retinal diseases. We're very pleased to have him here, and I'm sure he will give a very excellent and interesting talk.
Thank you very much, Sam. Let me see if I can share the screen here. And okay, let's see. So we tested it, Chris. We know it works.
It works. It's just you need to do the presentation mode.
Well, it's not sharing here. So we had the dog's head stuck in the door, and there we go. Can you see that?
Yeah, we see it. It's not in the presentation mode.
Yeah, but we have the little green bar.
Perfect, perfect.
Are we happy now?
Some of us. Tony, are you happy?
I'm very happy, Chris. Hi,
everyone. Okay, well, great. Thank you again very much for the invitation to be part of this great seminar series. And I don't have any financial disclosures associated with this talk. And as Sam said in the introduction, I've been very interested in the latter part of my career in sort of breaking down the barriers to getting treatments for these diseases to the patients. And as many of you on the call know, that a lot of the treatments that have started sort of have targeted price tags in the $1 million to $2 million range. And if you do the math on that, even for rare diseases, rare inherited eye diseases, that's just a prohibitive cost to the healthcare system. And when you imagine some of our the things we'll talk about today being applicable to more common diseases like age-related macular degeneration, the costs really need to come down. So our goals are to come up with ways that high-quality genetic testing can be delivered for less than $1,000 a person, including counseling. The paper that I circulated this morning has a discussion of this thing we call the false genotype rate. It's beyond the scope of this morning's talk, but it's basically the idea is this really widespread shotgun genetic testing is positive 128% of the time, and a lot of it is wrong. And so I'd like you to read about that in that paper. And we want to develop gene therapy for dozens of genes, be able to deliver that for $20,000 a person, including the surgery, including follow-up. And then the holy grail, of course, which you're very interested in at your institution, is photoreceptor replacement to actually restore vision to people who've lost it. And we need to get the cost for that down to under $50,000 a person. The keys to these outcomes, in our opinion, is to do a lot of the technology development in a nonprofit structure with all open source technology so that other people can copy it and drive the cost of these things down. The processes are super complicated. A lot of, especially the stem cell things, they have dozens and dozens and dozens of steps. And so unless every step is executed with a 99.9% reliability, you just don't have any product at the end of the process. As I'm going to show you, a very important thing is to deliver education about inherited eye disease very widely, because a lot of programs don't have very well developed genetic eye disease programs. A lot of residents and fellows just don't see patients with these diseases very much. So we need to deliver that educational material widely. And, you know, we need all these energetic optimism, creativity, et cetera, to make these things happen. So this is the paper that, that I circulated. We did this between 2012 and 2017, where we studied very intensively 1,000 consecutive families that I saw in my clinic at the university. And we did that basically to establish a denominator so that we would really know the relative frequency of all these conditions. We would know really how many genes were involved in them, and the answer is a little more than 100. And but we also paid attention to the clinical features that would allow a clinician to tell exactly what a disease was. And we've kind of captured all of that information on this electronic teaching website that we call Stone Rounds, because that's the name of the teaching conference that I hold here at the University of Iowa for the last 20 years or so. Once a week now, because of the pandemic, we actually hold that teaching conference via Zoom, and all of you are welcome to join that conference whenever you like. And that little schedule button down at the bottom of the splash screen, you can find out when this when they are. They're usually Tuesday nights at 5:30 Central Time. But if you were a frequent attendee of this Stone Rounds thing, you would be able to diagnose the 3 patients that I'm going to show you in the course of this talk. Not only the class of the disease would you be able to tell, but you would have a very good chance of knowing exactly what the gene is. And you'll see that it's important to know exactly what the gene is clinically, because in all 3 cases, the genetic testing was difficult, and one had to have a really strong suspicion of the disease to figure it out. So our first patient that I'm going to show you is the eldest of these 3 boys, and he's 7 years old, and he has 2100 vision, and he was 20/20 a year earlier. So is anyone bold enough to suggest what they think this child has based on just that story? So hearing none, I'm going to show you the fundus appearance. So you see that the fundus looks pretty normal, although, you know, frankly, the central macula is not quite as shiny as you would expect it to be. There's a little bit of wrinkling of the internal surface suggesting some photoreceptor loss. And then right in the center, there's a little sort of bullseye lesion. The Goldman visual field is somewhat reassuring in that it's full to the larger isopters, but the 1,2 isopter, 1,2E, shown in red there, for that to be lost at age 7 is actually quite concerning. And so this is the OCT tomogram of this patient. And it doesn't look too terrible at first glance, certainly not to people that aren't familiar looking at these things every day. But this is the clinical teaching point for this case. I'm going to show you a patient on the bottom that has bad Stargardt disease, which is what the presumptive diagnosis for a lot of these 7 and 8-year-old children are. And you notice the better lamination down here. You see the better internuclear layer that you can see. And this is sort of isoreflective. So when you see a 7-year-old patient who has had a very serious loss of vision over a 1-year period, and it has this isoreflective OCT, that's very suggestive of one very bad disease. Before the patient came to us, they had one of these popular hundreds of genes retinal dystrophy panel, which failed to yield any result. And the reason is that the patient actually has Batten disease, a fatal neurodegenerative disease of childhood caused by this gene CLN3. The most common mutation in this gene is almost 1 kb deletion. And shockingly, this commercial panel does not have an assay for that gene built in, and so that was missed. And then the other mutation was non-exomic. It was a mutation inside an intervening sequence, and we had to detect that by sequencing because we had this deep suspicion of what the disease was clinically, and then go on to prove that that thing 24 base pairs away from the edge of the splicing junction actually caused missplicing, which it did. So our second patient is a 38-year-old gentleman, and he is of Jewish ethnicity. He's 20/20, and he recently noticed some night blindness and reduced peripheral vision. And this is his fundus appearance. And so I'm going to give the audience a second chance here. Does anybody want to suggest a specific diagnosis for this gentleman? There are only 3 patients this morning, so so this is your second of 3 chances to use your electronic presence. Okay, hearing none. I will tell you that this patient has retinitis pigmentosa, but of course, you knew that. There's a very specific kind of retinitis pigmentosa that has this odd nasal predilection, which causes this very unusual deep cut in the temporal visual field. Most retinitis pigmentosa patients don't have that. So the combination of the normally sighted parents he has, meaning it's autosomal recessive, he has this nasal predilection and this deep cut in his visual field, and his Jewish ethnicity tells you that this person has a specific mutation, which I'll show you in just a moment. Here's his optical coherence tomogram. And for those of you who aren't used to looking at those, I put a normal one up here. This is the normal outer nuclear layer. You see they're dark. And he's lost his outer nuclear layer temporal to the fovea. So, so my dog, who I've rescued from the cat door, is now growling at the neighbors. So anyway, that's what's going on. She's being coaxed out of the room. Okay, so anyway, this patient, if you did a standard whole exome sequence on this patient, you would find nothing true. Anything you found would be unrelated to his disease. And the reason is that he has a 353 base pair insertion into exon 9 of this gene called MAC. And so the deal is normal exome sequencing protocols remove repetitive elements like ALUs intentionally, because otherwise all the sequence you would get would be repetitive elements. And so if the mutation consists of an ALU insertion, usually that element will be removed from the library and you won't be able to see it. But what you should know is this single ancestral allele accounts for 35% of RP in Jewish people. And so if you know the person is of Jewish ethnicity, they have RP, and then they have that nasal predilection, you can basically call this mutation on clinical grounds alone. And one PCR reaction will find the correct answer for the patient, even though exome sequencing would miss it. So the MAC gene is expressed throughout the retina, enriched in photoreceptor cells, and this is sort of what we might call the standard way of showing the expression of a gene. But I wanted to just acquaint you with this, this website that my partners Rob Mullins and Drew Voigt put together. They've been very active in the single cell RNA sequencing arena. And because Rob Mullins manages the research eye bank at the University of Iowa and gets lots of eyes with very short depth of preservation time, we have a lot of data sets from these, which we've made publicly available. And this so-called spectacle website, the singlecelleye.org, allows you to enter any gene that you're interested in and actually query the expression profile for that gene. So I encourage all of you who are doing experiments in the retina to use that website to look for expressions of the genes of your interest. This image just shows you that in our patient, there is no mature MAC transcript, which there's a unusual 25 base pair exon 12, 25 codon exon 12, and the mutation alters that isoform of the message such that it's lost, and that's the mechanism of the loss of the MAC function in these patients. So what MAC does for a living is it regulates cilia length. And so what you see here is just in fibroblasts, because all cells in the body have a primary cilium. In fibroblasts, you see that the mutant MAC fibroblasts have a very, very long primary cilium compared to normal, and that's easily quantifiable. So that just gives us a nice in vitro assay, if you will, of the gene function. This is a normal AAV packaging construct where we put the MAC coding sequence behind two different promoters, and we had these two different isoforms. We made the retina isoform with that extra 25 codons of coding sequence in it, and then the MAC isoform that's present in other cells in the body, because a priori, we weren't actually sure which one would perform better as a gene therapy construct. So I'll show you that we were able to restore both the canonical and the retinal protein in patient-derived retinal precursor cells. And this is the thing that we haven't fully explained yet. We use the two different promoters, CMV and EF1 alpha. The latter is drives the gene less than the CMV promoter, which drives it as much as possible, because we wanted to have a couple of choices of expression, because some genes exhibit overexpression toxicity when you're trying to use them as a therapeutic vector. What we were surprised to find is, by some mechanism that we haven't completely figured out yet, that 25 base pair the 25 codon addition in the retina isoform causes it to be translated dramatically more extensively than the non-retinal form. And what you see in the panels below there are just the expression of the retinal isoform driven by CMV and the retinal isoform driven by EF1 alpha are both more than either of the canonical isoforms. And happily, none of them exhibited overexpression toxicity. And in our early days of AAV-mediated gene therapy, that's really what you want, because what you'd like to do is put a strong promoter in front of the gene so that when you put this into the subretinal space, you can reach a lot of cells with it. But then when the cells take the vector up and start expressing it, you don't want the therapeutic protein to actually injure the cells. And so happily, in this case, it does not. Here, we just use the in vitro fibroblasts as an initial assay to show that we were able to restore the normal length of the primary cilium with the this construct. And then we created a rat model of MAC-associated RP. And on the left panel, you see a 4-month-old animal that's untreated, and you see that basically the entire outer nuclear layer is gone. And in the 4-month-old animal that had been treated 1 month earlier, and why did we do that? Well, obviously, the earlier the better. The earlier the better, because you want as little retinal death as possible for the maximum therapeutic effect. But we wanted to just sort of get the idea that if we had a patient who had already lost some photoreceptor function such that they presented to us clinically, would this therapy potentially slow their disease down? So we intentionally treated a 3-month-old animal that already had some photoreceptor death to see if there was a therapeutic effect, and there was. And so the combination of this favorable effect in the animal model, plus the lack of overexpression toxicity, encourages us that we can go ahead and move forward with this in human beings. But my question for you is, what if that rat experiment had failed? What if the rat experiment had failed? Meaning, you know, the rat disease is pretty severe. It's a lot more severe than the human disease. The human disease doesn't even get diagnosed until somebody's in their late 20s or early 30s. And this rat disease, inexplicably, is really severe. So what if it had failed? Would we not have gone forward? Well, we would argue that that would be a mistake. There are lots of animal models that don't mimic the human disease very well. And so we think that if you can show that you can restore the normal protein to human cells, and that there's no overexpression toxicity either in those cells or in a normal animal, that's pretty good evidence for going ahead with a one-eyed clinical trial in human beings. That's not the standard right now. The FDA generally expects an animal model, but we think that's a mistake. We think that you should be able to simply show that the expression is safe when it's overexpressed, and that you can restore the normal protein. If you do that, and if you execute the manufacturing and the clinical trial in a nonprofit fashion, we think we can get down to those low costs that we described earlier. For example, if you have your own GMP facility, which we do, that was built for us by philanthropists, you can make enough clinical grade AAV to treat 500 people for less than $25,000. If you have your own veterinary pathologists and animal facilities, you can do all the preclinical toxicity stuff in-house in less than 6 months for about $200,000 per target. If you can talk your hospital into letting you do the surgery at costs, you can do a clinical trial for about $20,000 a patient, including 10 years of follow-up. And what all these things mean is a relatively modest sized group like ours can go after 3 new genes a year and make enough of the stuff to treat everybody in the United States while doing so. And then obviously you would have a network of institutions that would collaborate to do the surgeries and so forth to really get these treatments out to a large number of people for a small cost. So as you probably gathered, CLN3 and MAC are two of the first genes that we have worked on and gotten pretty far along in our affordable gene therapy pipeline, and hope to get those into human clinical trials soon. So here's our third patient that I'm going to show you today. Also a man of Jewish ethnicity, and he's got 20/25 vision. His vision is right eye is poor because he had a previous retinal detachment. He has night blindness and reduced peripheral vision, sort of a similar age of onset as our other patient. Here's his fundus exam, and you see that he doesn't have that nasal predilection. So I had you all tuned up for this Jewish man thing, so you're going to go for MAC, but he doesn't really have the nasal predilection thing, and he doesn't have that very unusual cut in the temporal field, which is a feature of that retinal predilection. So what should you suspect here? Anybody want to say what gene they expect here? Third and last try. Okay, the most common gene that causes autosomal recessive retinitis pigmentosa is USH2A. So the gene that causes type 2 Usher syndrome actually is frequently a cause of non-syndromic autosomal recessive RP. And here, like the other two patients, this guy has one of his mutations deep, deep inside an intervening sequence. And that was actually difficult to find. And once you find it, you need to actually prove that it's disease causing, which we did in the patient-derived iPS cells. And so in all of these three cases, if you had done just the standard 250 gene panel kind of thing and left it at that, what you would have found was a lot of other plausible disease-causing variants in other genes that weren't actually the cause of these patients' disease. So again, that's the false genotype rate thing that I told you about. So here's our patient who is very happily wearing his Institute for Vision Research t-shirt. And if he looks at himself in the mirror today in his 60s, this is what he sees. So he's down to just very, very small amount of visual field. So obviously gene therapy isn't going to help him. In order to get some meaningful treatment for him, we're going to have to put some cells back in there. So here's a schematic of the normal retina. And so in retinitis pigmentosa, all of the outer nuclear layer and the other structures, the photoreceptors are lost, the retinal pigment epithelium is injured a bit, and so forth. But happily and somewhat miraculously, a lot of the inner retinal structures, the ganglion cells, are spared. And that's fortunate because these axons go several centimeters from the retina down the optic nerve to the lateral geniculate nucleus in the brain. And if those were injured, I don't know exactly in 2021 how we would fix that. But fortunately, because all this wiring is still intact, the idea is to just put some new cells of photoreceptor lineage under the bipolar cells and hope that they will form synaptic connections and subserve vision. And this has actually, this was work from Bud Tucker in 2011, but a number of other groups have shown some restoration of electrical activity in the retina. And it's not just the A wave, if you will, there is a bimodal response to the ERG suggesting that there is some actual synaptic connectivity there. So that's what's really, these kinds of experiments are what are really driving everybody's hope that we're going to be able to restore vision with these photoreceptor graphs. So when you start imagining doing that, there are really kind of two strategic decisions that you need to make. One, you need to decide whether you're going to make the cells you're going to transplant from the patient themselves, or whether you're going to use a bank of cells that are not derived from the patient. And the second is whether you're going to support these cells with a polymer, or whether you're going to just inject them into the subretinal space or the vitreous cavity. So we've decided in our group to focus entirely on autologous cells and to support them in a polymer sheet. So why autologous? When you transplant retinal allografts in pigs and primates, there's a very significant immune reaction to those unmatched cells. And that's true even when the animals don't have prior retinal disease. So you could potentially get around that with, immunosuppression. Certainly that's been used extensively in other human transplantation, like the heart and kidneys and so forth. But we know in those types of transplants, the more well-matched the tissue is, the longer lived are the grafts. So cells derived from the patient from whom they are intended have a perfect immunologic match. And so it's possible that there would be a little bit of foreignness to them because of the manipulation that would be involved in creating the grafts, but they would be much better than any sort of allograft. So as you know from the work that is done at your institution, iPS cells can be used to make very, very authentic photoreceptor precursor cells these days, and they are suitable for transplantation. The problem with the autologous ones is that they would still have the genotype that caused the person's disease in the first place. And so that's a problem. Fortunately, the CRISPR-Cas9 genome editing technology that sort of evolved very shortly after the iPS technology allows us to correct any mutations in the patient's cells at the iPSC stage, and then you can sequence them and make sure that you haven't created any other untoward mutations somewhere in the genome. And so then what you have is you have the patient's own cells, and the gene that caused their disease is normal and corrected and under the normal promoter. So the way this differentiation looks, you start with fibroblasts and treat them with the 4 Yamanaka factors, which results in these iPS cell colonies that are, basically just like the cells shortly after fertilization in a human embryo. These cells are then subjected to a differentiation protocol in which growth factors are adjusted every day or two to mimic the environment that these cells would experience in a normal developing baby. And what you see here is by day 16 in this differentiation medium, you get these optical vesicle looking structures, which are then lifted off the plate and grown floating in culture. And when you do that, you basically get these retinal organoids by day 30 that basically have a palisading group of cells that are sort of developing and maturing outwardly so that the more mature cells are on the outer surface of the structure. If you continue growing these cells in a differentiation medium for another 100 days or so, when you get to 150 days, you have very, very mature photoreceptors in these organoids. So here I've just magged up on this part of an organoid showing cells that are expressing medium and or long wavelength ops, and the antibody can't tell the difference between those. But you can see that little cone outer segment, very normal looking cone morphology. And then rhodopsin expressing cells with this beautiful normal looking outer segment there directed outward, directed inward in the layer there. So the problem with these cells is that if you dissociate them and just suspend them in tissue culture medium and inject them as a bolus under the retina. A few of the cells will survive, and that's how the initial experiments were done, but the vast majority of them won't. So maybe 2–3% of the cells survive. If you can support these cells with a biocompatible polymer, 85–90% of the cells will survive. So the next sort of frontier then is to develop the polymer support to make it optimal for this purpose. And so we've been using 2-photon lithography for this purpose. This is a just spectacular technique in which you can print features as small as 75 nanometers. Here you see one of our early attempts at printing one of these scaffolds, and we had the idea that we called this the egg crate because the holes were about 10 microns. And I think we were so enamored at the idea that we could actually print 10 micron holes that we wanted to make those holes as small as we could so that each photoreceptor would be down at a hole. That, of course, was not very thoughtful, because we ended up with way more polymer in this thing. You actually want as little polymer as possible to support the cells. But also the cells like being together more. They don't want to be quite that isolated. So the current scaffold that we're using is a more open architecture that has these sort of lobster trap looking wells with a porous bottom. And the newest modification is that we've added this little gap here at the bottom that we imagine that the outer segments are going to be able to protrude through and not be crushed against the surface when we transplant them into the subretinal space. This just shows you that we can actually make 5mm diameter versions of this now. And when we load them with photoreceptor precursors, you see that we can get very, very full loading. This just shows a couple of cone markers to show that there are a lot of cone photoreceptors in there, which is desirable for therapeutic targeting to the macula, where we would hope for these cone lineage photoreceptors to wire themselves up to bipolar cells for good acuity vision. So our initial experiments were imagining in humans we can get 2 million cells loaded onto a 5mm graft. And so we imagine putting that right into the macula of completely blind people. We're going to treat completely blind people because obviously we don't want to injure the vision of a sighted person with unproven technology. But also it'll let us, really believe the result if we get some sort of formed vision back from a blind person. We've performed this surgery in large animals. Pigs, as you know, have eyes about the same size as human, and so that allows us to test the surgery repeatedly in large animals. And here you see the biocompatibility of this structure under the retina for a month in an animal that has retinitis pigmentosa. So you see these cells of photoreceptor lineage tolerating that graft for a month. But for FDA work, they want to see hundreds of animals, and they also want us to look at all the organs in the animal to make sure there's not some remote experiment. So we put iPS-derived photoreceptor precursor cells from 5 different individuals on grafts and put them each into 30 animals and aged them out. Here's Ian Hahn putting these 1mm grafts into the subretinal space of rats, which I just find absolutely amazing that that can be reliably done. And you see here after 6 months, the graft is still in the subretinal space, has not eroded. None of these CRISPR-corrected cells have formed tumors, and which is a concern that people have whenever you're using induced pluripotent stem cells. And So anyway, that's where the state of that retina only transplant program is, the photoreceptor only. But what about multi-layer grafts? If you can make photoreceptor layers, what about going ahead and building a whole outer retinal structure? So obviously you can make really, really nice retinal pigment epithelial sheets. This has been done for many years, and those can be grown on porous surfaces. But one of the coolest recent things is we've gotten to the point where we can actually make iPSC-derived choroidal endothelial cells through a CD31 pull-down enrichment strategy. And if you seed these endothelial precursors into matrigel, you see that they'll actually form these vascular structures. They're actually tubes that look actually quite normal. And you can modulate the periodicity of these vascular structures by altering the oxygen tension of the culture medium. So we're experimenting now with polycaprolactone surfaces for these choriocapillaris, artificial choriocapillaris layers to grow on. And so here's an example in which we have RPE cells growing on a millicell insert, sort of as a pseudo Brooks membrane, and then that's on top of one of these choroidal endothelial cell structures. And the whole thing is in a perfusible incubator system in which you can actually have tissue culture medium perfusing underneath the graft, simulating blood flow. So all of this is to allow us to experiment with the different growth factor interactions between endothelial cells and RPE cells. But then, of course, you can put one of the polymer-supported photoreceptor structures on top of this thing and have a full three-layer outer segment structure. So how do you make enough of these CRISPR-corrected autologous grafts to treat hundreds of patients in a single institution? Well, that's going to require robots to do, because human beings just cannot do all of the steps that are necessary, especially spanning 150 days of manufacturing. You just can't do that flawlessly, reproducibly enough, all with people. And so we have been working on different robotic helpers, so to speak. This is one that can actually identify iPS colonies as well as our best technicians can. And the robot can come in and pick those iPS colonies and transplant them into tubes for further work. And then, you know, the real killer is when you're trying to feed these cells different types of differentiation medium spanning, 150 days. Well, if you were going to do surgery in 2 patients a day, you can't have all of them coming due at once. So you have to keep track of all the different patients and where they are in the differentiation protocol and so forth. So here you see this Lyconic incubator, which has a carousel in it that can hold 500 different patients' cells at a time. And then a robot can reach in and pull these patient cells out one at a time and put them on that feeding robot that you see in the back. And then, of course, it's got barcodes on the plate to tell you which patient it is. And then it gets exactly the right differentiation medium for that day. So in summary, I've shown you that as genetic tests become less focused, clinical knowledge is more important than ever. So a lot of people think that, now that we've gotten to the point that you can do exome sequencing and everything, what do you need clinicians for anymore? And I'll tell you that it's actually the reverse. You need more clinical expertise in the face of all that genetic polymorphism to get the right answer. So please, those of you who are clinicians who've been interested in inherited eye disease, check out the Stone Rounds website and consider joining us on Tuesdays for our teaching conference. I've shown you that the keys to affordable gene therapy are going to be doing the manufacturing and clinical trials in a nonprofit environment. And we think that we need to, push forward with acceptance of in vitro models so that we can treat diseases that just don't have a reliable animal model for the human disease. Photoreceptor replacement is getting closer and closer to the clinic. We think that using autologous cells are going to reduce the need for expensive and toxic long-term immunosuppression. And as we get better and better with our robots, we're going to be able to keep the manufacturing costs down and increase access for our patients. So I want to, thank the team of people that work together here in the Institute for Vision Research. Obviously, this huge body of work that I've shown you today represents the work of 75 people spanning really decades. But I want to highlight one person for sure, and this is my colleague Bud Tucker, who is responsible for lots and lots of the work I showed you today. All of the stem cell work, the design and running of the GMP facility, Bud does that, and all of the FDA work as well. So with that, I will pause and be happy to take questions.
Thank you very much. This was really fantastic, unbelievable. If you guys have any question, please send me a note by chat that you have a question to ask. And we will give 1 minute to hear from you.
Well, Ed, I'd just like to say, as a clinician scientist not in this field, I'm blown away by the progress that you've made. It is amazing, and it's a blueprint for the future for so many people, I think. And you've taken that into consideration in developing your technology. Congratulations.
Thank you very much.
All right, let's go to Dr. Lewin.
So, Chris, Dr. Stone, that was really a wonderful talk, very, very clear and very encouraging. I wonder if you've used any of the organoids to test gene therapy. I think the FDA will be willing to approve therapies when you go that step if they appear therapeutic in an organ-like structure in vitro.
Oh, yes, absolutely. And I didn't have time to talk about that branch of the work, but obviously having access to these living cells enables you to develop pretty sophisticated in vitro phenotypes for a lot of these diseases. And so the idea would be able to correct the surrogate in vitro phenotype in the patient's own cells. And we have done that with a lot of different diseases. One problem is, though, that if you had a fairly subtle disease that might not manifest until you're 25 years old, that might not actually have a really detectable phenotype in cells. And so the point I was sort of making was, in the cases where you simply can't discover an in vitro phenotype, or you can't really develop a reliable animal model, but you can show that the construct isn't toxic and it restores normal levels of the normal gene product, it seems like in the absence of any other available treatment for this degenerative disease, that a trial of putting that into one eye of a person who still had some photoreceptors left would be preferable to not having any treatment under trial. And so that's sort of our pitch on that. If you have an animal model, fantastic, use it. If you have a good in vitro phenotype, absolutely correct it. But when you don't, and we don't for a number of important diseases, we think it's reasonable to go ahead and make a construct and start putting it into one eye of a person.
All right, Yoshi.
Thank you very much, Dr. Stone, for a great talk. I have a question about your RPE cell culture, in which you utilize Brooks membrane type of material. So my question is that, is this what you also created by two-photon lithography, or do you have different kind of design principle for RPE versus photoreceptor cells?
Yeah. So, it's so funny. This talk has 87 slides in it, and like, I don't know, 1,000 words and labels. And the one label that I was going to change was I was going to change that BM on that slide from Brooks membrane to MCI for millicell insert. So we made that figure, and when we made that figure for a talk, and we said that this was an artificial Brooks membrane, so to speak. Really, all that is right now is a standard millicell insert that's porous, that the RPE cells are growing on. But in the future, we would make, try different dissolvable polymers and so forth for that layer, because I don't think you'd want to actually transplant that millicell material into a person. But for the purpose of studying the interactions between RPE cells and endothelial cells under them, that porous millicell insert is a perfectly good starting place. But it should not have had BM on that slide. It should have had some sort of acknowledgement that that was actually a millicell insert.
You see, Ed, Yoshi will never let it go if there is something wrong, and so.
Actually, there's another thing in there that we'll see if any of the questioners get. But you got the one that I didn't manage to get changed. So
okay, Yoshi, you work harder. Today we have a very, very good news. Dr. Swarup will make a final comments and final question. But before we do so, let's go to Zach.
Hi, Dr. Stone. That was a great talk. So I have some questions, or a question about the seeding of the photoreceptor precursors into the retinal space itself. So those are rigid scaffolds from 2D lithography. Do they actually conform to the curvature of the retina? And also, like, in terms of injecting or inserting these scaffolds, I mean, are there shear stressors that could possibly affect cell viability when you're injecting, for example, like freely solubilized or freely in solution photoreceptors that have been dissociated from organoids? I'm just wondering if there's like better methods of not only promoting these photoreceptor precursor, the photoreceptor precursor viability, but also increasing distribution across the entirety of the retinal space.
Well, this question is a very important one, and the physical properties of the polymer are extremely important. And some of the initial polymers that were used in this field were too stiff. And so we called them cheese graters. So if you made little holes in stiff material and put it under the retina, then in the normal movement of the eye, it just shifted this thing back and forth, and it eroded, just chewed up the retina. So you want the polymeric material to have a bulk modulus that's basically identical to the retina. And this polycaprolactone material is like that. So that if you imagine the floppiest soft contact lens you've ever held in your life, that is what this thing looks like. And it looked rigid when I was showing you the pictures because it was scanning EM, and so it looked like a cinder block. But the material is actually, really, really soft and pliable, and it's actually dissolvable. It actually dissolves over time.
Okay.
So the way you transplant this thing is you load it with the cells, and the cells grow in the planar structure for a while. But then to put it in the subretinal space, you sort of use a trick similar to foldable intraocular lenses, and you roll this thing up in a little injector, make a small retinotomy temporal to where you're going to put it in, inject some BSS under the retina to form a bleb, and then you push this thing through the hole in the retina, and it flops open on the other side like a contact lens. And then, yes, it does conform. And so that's why I was showing you these animals where these things have been in the subretinal space for months, and the retina is just perfectly happy on top of them. So the bulk modulus is a super key feature of this technology.
So would it be possible then to kind of do like a micro mosaic of these scaffolds?
Oh, absolutely. Absolutely. But yeah, but the deal is, as you know, with any kind of surgical procedure, if you have a difficulty of 1 for putting 1 in, you have a difficulty of 2.5 for putting 2 in, and a difficulty of 5 for putting 3 in, right? But so initially, if you got a 5 millimeter graft in there and it worked, meaning the whole surface of it formed synaptic connections and everything, that would result in a field of vision that would be, 30 degrees wide or something. It would be fantastic compared to a patient that had no light perception vision. But then as you're imagining the future, you could think of putting one of those things superiorly so that you'd get a patch of vision inferiorly, and one of those things nasally so you get a patch of vision temporally. And just each one of those would make the surgery a little more complicated and a little more likely to have a problem. But, when you start giving surgeons stuff like this, they'll be off to the races with umpteen different designs and shapes and fitting them together and all that. It's just the 5 millimeters in the center is the place to start.
All right, let's move to Jason.
Thanks, that was just a terrific talk. I wanted to get a sense of how long photoreceptors in organoids, relatively mature photoreceptors in organoids, will survive after dissociation, and what steps need to be taken between the dissociation and the seating on the scaffold to encourage survival, exactly because photoreceptors want, neighbors in order to survive.
I don't know if we've ever really done that experiment. In other words, the seeding, you don't maintain the dissociated cells for any period of time before you seed them. You dissociate them and seed them. And so then fairly shortly after they've been dissociated, they're back in a stable place, touching each other and so forth. So I don't know how long they would survive if you attempted to keep them apart. Probably not very long, because that's the whole anoikos phenomenon. That's probably why the bolus injections under the retina don't work very well, because the cells, of photoreceptor lineage don't like not touching their partners, and they'll apoptose if you do that. But anyway, the seeding is done by taking cells that are together in the organoids, dissociating them, seeding them right immediately onto the polymer, and then they're all, packed in together again, but they're just supported now by the polymer.
All right, Iris. Looks like we lost her. How about Emma?
Hi, Dr. Stone. I'm Emma. I'm Dr. Karen Pusak fellow, so I'm interested on the retinopathy and the vasculature. So the question that I have 2 technical questions. The first one for the your choriocapillary sheets. You show them that you put it in an horizontal plane. Have you been able to put them in like a hollow tube this position, so you can induce flow through the hollow or matrix, so you can influence them by this type of flow rather than a laminar flow just in an horizontal plane that you're showing.
Yeah, well, the two things that we have done with them so far, I basically showed in the slides. One is they will grow sort of multidimensionally in matrigel. And so if you take the cells and seed them in matrigel, they'll form these growing networks. And so if you were going to do experiments like what affects the branching and you know, what kinds of growth factors would make them be fenestrated versus not, and things like that, you can get these sort of networks in matrigel. And so then the other one, the planar thing, is we said, well, you know, the choriocapillaris is more flow in a plane, not flow in the structure of the liver, you know. And so we wanted to sort of constrain them physically by giving them the surface to grow on. And then we have this tissue culture medium flowing by them, not through them, you know, but by them. And the idea was to just mimic as closely as we can the physics of the behavior of the choriocapillaris, so that the retinal pigment epithelium is exposed to whatever growth factors are elaborated from the endothelium. So it's thinking it's, you know, because there's this concept of the photoreceptor RPE choriocapillaris unit. And if you injure any one of the three, the other two are lost. So they're clearly talking to each other, you know, in some way, and yet the nature of that communication isn't well understood. So the idea is, by having this 3-layer graft system, you can manipulate things in either one of those, in any of those 3 layers, and see what it does to the other layer. But to go sort of, I think, where you're going in the transplant dimension is like, well, what would happen if you transplanted that into the subretinal space of a human? You know, would the endothelial cells do anything? Well, as you know from diabetes and other diseases, that microvasculature remodels all the time. So what if you put that thing in and it had formed all these tubes and everything? Would it actually know how to anastomose to the edges of healthy endothelium? Maybe it would, or maybe you could make it so. I didn't mention this other thing, but this is a great opportunity to do it. The other thing about polycaprolactone is you can actually embed growth factors within it, so that it becomes sort of this slow release drug delivery system, for want of a better word. And so with the 3D printing, what you can do is you can be printing along and then change the bath and put a growth factor in, so that it is embedded in just one surface of the graft. And so if there was a specific growth factor that would promote vascular fusion, for example, you could put that on the surface of the graft and have it be released for, you know, 3 weeks time or something, and then go away to maybe, you know, and so you could also have something like that for synaptogenesis. Maybe the bipolar cells don't really want to form synapses because they're mature neurons, but you could put a growth factor in to drive those bipolar cells backward in developmental time and make them more plastic. But then you wouldn't want them to stay like that forever. You'd want them to return to the mature state. So you'd want that growth factor to expire. The cool thing about all these different systems is it's just going to give us the opportunity to test all of these different dimensions of biology in a way that we just haven't been able to in the past.
All right, let's move to Susie.
Thank you.
Hi, Dr. Stone. Thank you for a very inspiring talk. I have a question. Could you elaborate a little more on your approach for genome editing in patient-derived cells? So, for example, do you deliver like a Cas9 protein with the template and guide RNA to promote homology-directed repair? And if you do this way, how do you sort out the correctly edited cells among all other edited, other like modified cells?
This is such a great question, Susie. Thank you for that. So, you know, obviously CRISPR-Cas9 is a big deal, and just, you know, arguably as transformational methodology as iPS cells themselves, and give us just this tremendous ability to manipulate the genetics of cells. But the downside of them is they're not very efficient. And so the reason CRISPR-Cas9 isn't the solution for all gene therapy is that if you put these CRISPR-Cas9 reagents into the subretinal space, let's say, you'd only get a few % of cells corrected with that. The beauty of CRISPR-Cas9, though, in tissue culture is you have the luxury of cloning the result, right? And so all you have to do is correct the iPS cells and then clone them, meaning grow them at clonal density, pick clones and sequence them looking for the corrected cells, and then you grow a new culture up from the cell that is completely corrected, and then you have 100% pure clonally corrected cell line. Then the other concern that people have is that obviously if you put a, you know, CRISPR-Cas9 machinery into a cell, it could have off-target effects that would be untoward. And so you want to make sure that that hasn't happened. But it's just, as a person who came up, you know, in my scientific career starting in, you know, 1986, the idea that you can sequence the entire genome of a human being for less than $1,000 now is just, you know, shocking to me. But you can. And so now, after you've CRISPR corrected the cell line, you can do whole genome sequencing. And, you know, you're not going to find every single little piece of lint in the genome there, but you don't really care about that. What you care about is that you haven't inadvertently introduced a potentially cancer-causing mutation in a known cell cycle gene or something like that. So in summary of all that, CRISPR correction in iPS cells, clonal selection for cells that have the correction that you want, followed by whole genome sequencing of the cell line after that to make sure you haven't induced any untoward genetic injury.
Okay, we have wonderful conversation, not only presentation, but conversation. Let's limit to 2 more questions and then we go to Anand. So Ben.
Yeah, good talk. So I have a question. I work in Madeleine Siler's lab. We differentiate the human employed stem cell into the retinal organoid, and then we cut it in the small sheet and then transplant it in the subretinal space to try to restore the vision. So I'm curious, for your way, you use the polymer. Have you tried to just directly cut your organoid in a sheet and then transplant it inside the animal model and compared to the polymer? Do you need the polymer?
So I'm well aware of the work of your laboratory and congratulate you on that. What we think is that we have better control over the cellular composition that's actually in the sheet with this method, and the anatomy is more favorable. The more uniform anatomy is more favorable. But, you know, certainly I think organoid-derived sheets would be definitely preferable compared to dissociated cells in a bolus, which is what the original thing was, because the cells in the organoid-derived sheets, in my view, would have a much greater chance of survival and integration than individual suspended cells shot through a needle. But we think that the addition of the polymer support and the ability to deliver exactly the cell composition that we want to the surface of the polymer is optimal, which is why we're using that.
Josiah.
Dr. Stone, thank you so much for an amazing talk. That was wonderful, truly. My name is Josiah. I'm one of the postdoctoral fellows in the Andrew Brown lab. And as an engineer, I'm pretty interested in your 2P 3D printing. So I hope you don't mind if I ask you some questions on that. Two questions. The second one's pretty related to the first one. But the first question is, what are the factors that you were considering when choosing the materials for your 3D printed scaffold? Like you mentioned, the bulk modulus was one, maybe growth factors, maybe the fact that it dissolves potentially. And then the second question I had was, what are your limitations of using that material? And I guess also similarly, what are the advantages of it?
Well, that question would have a 2-hour-long answer. You probably are aware of that. So, obviously we can't do that. I will tell you that, first of all, kind of the design considerations are that some polymers that have favorable physical properties are actually toxic as they break down. And so as the scaffold depolymerizes, many polymers actually have an acidic byproduct as the monomers are released. And if that happens too rapidly, it's actually pretty toxic. So the polycaprolactone is just one sort of open source polymer, which has that advantage that it's freely available and can be used by anybody for any design they want. And then it has these favorable biocompatibility profile. The second one, a second design consideration is that for this nanoscale 3D printing, you need to be able to covalently link a photoinitiator onto the monomers. And so there's a little chemistry involved there, and not every single polymer would be equally amenable to that. And then the third one we touched on already is this, the bulk modulus of it. There is, as you might imagine, an intrinsic bulk modulus to the material. That is, if you had a whole block of it, what would be its bulk modulus? But you can also adjust the bulk modulus of the graft itself by making the little holes bigger and smaller. And so you might have noticed, being the engineer that you are, the little cuts on the sides of the lobster traps. If you made those cuts wider, the thing would be floppier. And if you made the cuts thinner, the thing would be stiffer. And so ideally, you would want it to be just like water, but then the surgeon couldn't handle it. The surgeon would like it to be like a piece of plastic, because he or she wants to be able to grab that thing with a forceps and stay right where they want to put it and everything. So the balance is, can it be floppy enough that it won't erode through the tissue, but have enough integrity that the surgeon can actually manipulate it and put it where it needs to go. So that's my short answer to a very good question.
All right.
Thank you so much. Appreciate it.
Expectation. Let's go to Dr. Swarup with all expectations.
Yeah, thanks, Chris. Yeah, Ed, some of us who have known you for quite some time, since very early days, I think we have always admired your very ambitious vision and goals. And today was no surprise. It's just amazing that you have been able to accomplish some of your dreams as well. I mean, I still recall some of the early talks you and I had in Michigan, and I'm amazed. I'm truly impressed, and thank you for all the stuff. I just have two comments actually. So one of that is that your use of polymer embedded photoreceptor sheet or those sheets of cells, I think is a wonderful idea, because it's not only going to take care of this the cytoplasmic transfer issues that people had, but also, I mean, I personally never liked those squirting the cells in a needle and all that. So I think it does give support, and I think you have good chance of success, and combining it with autologous cells after CRISPR. I mean, I hope you succeed. I mean, I hope you really, I mean, that's, and you can deliver that in a small enough price that you're talking about. I mean, I think that's the bigger goal that you have set for yourself. So I think I'm truly impressed and very, very happy that you're doing it. Second thing I wanted to comment on was your statement, which I have tried to tell a few people, but I'm not a clinician, so who listens to my comment, but I think your comment I'm going to use, if you don't mind, is that animal model, even if you don't succeed in that, if somebody can show therapy in organoids or by some other means, I think it should still be tried. And the reason, a simple reason for me, is that in many animal models, natural history may be different. And particularly for developmental genes or transcription factors or whatever, things happen so fast in mice and rat, and it's very hard to inject or take care of the right moment when you need to do the therapy. Whereas in human, the whole development and differentiation takes place over a very extended period of time. And even much later, you have many cones, even though they're dysfunctional, are present in macular region or whatever. So I think mice are not human, or rats are not human. And I think you're absolutely right that if one can show a therapy in an in vitro system that works, and you use some delivery sort of paradigms where there's less immune response, why not? Why do you have to show it in animal models? I mean, they may or may not work. And so, I mean, I sort of thank you for suggesting that. We have been facing something like that for CRX therapy. We recently published in Organoids, and people were saying, oh, why don't you do this animal model or that animal model? But I think I'm going to use your name now. Say, Ed said that it's okay. You should try. So thank you very much.
Well, Anand, thank you for those comments. And I would like to respond to both of them, and I'll do them in inverse order because at my advanced age, I'm not sure I can remember both of them. You might have to remind me of the first one. But anyway, regarding this this going ahead in humans thing, it shouldn't sound cavalier. Okay, nobody's talking about not doing safety studies. Nobody's talking about not doing safety studies. We're talking about doing all the safety studies in the usual way, showing that the construct is not toxic in normal animals. So you treat hundreds of normal animals and show that the construct does not hurt the retina. And then the only question is, how efficacious is it going to be? That's the question. And we don't know because we don't have a good animal model, or the animal model doesn't represent the disease well, because here you see this Mac rat. It gets RP all right, but it gets this terrifically aggressive RP. But in humans, it's a much milder disease that appears in adulthood. So how would you go about it in a human? Well, I showed you that nasal predilection, the nasal predilection. We intentionally destroy that retina in the clinic all the time in diabetics. Okay, nobody's standing around out there wringing their hands, when we're going to put 1,000 laser spots over there in the nasal retina. So why can't we put 300 microliters of a AAV construct in the subretinal space of somebody who's going to lose that retina from their disease? We know they are, okay? So we know the pattern, we know the disease is going to affect that. So how do we treat them ahead of time? Here's the beauty. MAC-associated RP is the most common cause of RP in Jewish people. Many Jewish people undergo genetic testing when they get married to avoid Tay-Sachs and other things like that. So if you test for MAC at the same time you test for Tay-Sachs, so that somebody doesn't want to have a child with MAC, what you will find is you will trip over some 22-year-olds who, unbeknownst to them, are affected. Okay, they're not going to have RP for 10 more years, but you're going to find them when they're doing their adult testing avoiding Tay-Sachs. So now you go put this construct in the nasal retina, okay? And then you follow them for 10 years. That's the other thing. All of the corporate models are so driven by the burn rate of their company. They want an outcome measure in 12 months or 24 months or something. Well, thankfully, these diseases don't go that fast. These diseases change over a decade. So what you want is you want to put it in a safe space. If, God forbid, the construct injured the retina, well, I'm glad we didn't put it in the macula. If, on the other hand, that eye, the one eye you treated, using their other eye as a control, is spared, and you do that in a small number of patients, 15 patients or something like that, then I would happily put it in the macula of one eye and follow them for longer. And that's the stepwise way. But if we don't even put it in any human being for 15 years because we keep casting about for a rodent model that's going to mimic the human disease, then we're just not going to treat anybody. Responding to the other comment about what we've been able to accomplish over the years, I just have to say again that this work that I showed today is the work of dozens and dozens of people spanning decades, and I want to emphasize that point. But you said, I hope that you will do it. Well, Anand, I will tell you that we have already done it, meaning we have already made clinical grade vector enough to treat everybody in the United States for $25,000. Okay, we've already done that. That has been accomplished.
Wow.
And so I was telling somebody this, because we have our own GMP facility that we built with philanthropic dollars. We did that. And so, and we have all the blueprints and everything. It's all open source. We'll share it with anybody. And I was telling somebody about this one day, and they said, well, Ed, what if somebody steals this idea from you? And I was momentarily, stunned by the question, because of course that's the entire idea, okay?
Yes.
Is for someone to steal the idea and do it. So that if we did 3 genes per year, somebody else would do 3 genes per year, and somebody else would do 3 genes per year. And in a few years, all 75 of the genes in the 1,000 consecutive families paper that will fit into AAV would have been made in a clinical grade fashion and tried in human beings.
Yeah.
Okay. So the whole idea is not that we would do it all, but that we would just show that it could be done at that price point. It could be done at that price point. And I frankly think it has to be done at that price point, because Batten disease, fatal disease in children, at any given moment, there are only 50 children in the United States that are at a point
nobody
in their
will be
disease.
able to afford $1 million a part. And I couldn't be happier if some of those companies go bust with all that, I mean, $1 million a shot for every patient. I mean, that's just
the problem is, Anand, is that regardless of, I tell people I'm not allergic to money or something. This isn't some sociological statement. It's just that our healthcare system does not have that money in it. It simply doesn't have it in it. And so if you were going to say that you were going to make a graft for $1 million, you were going to make a graft for $1 million or $2 million and treat atrophic AMD with it, well, multiply 1 million patients times $1 million and tell me where that money is in our healthcare system. It does not exist. So if you can get the cost of these things down to the cost of a knee replacement, well, now we're talking. Now anybody could get it. So I just want everybody to internalize those numbers. $20,000 for gene therapy, $50,000 for stem cell therapy. It absolutely can, CAN, be done in 2021. It can be done already. And now it's just a matter of everybody, you know, pushing forward and doing it.
All right, I will push a mute button so Dr. Swarup will not ask any more questions. I thank you all and going to see you next Friday, the same place, the same time. We will have another speaker, and tomorrow is another day. That's what I say.